It's my pleasure to introduce this evening Dr. Jason Hilton. Jason did his first degree in Sheffield University, before moving on to do his PhD, I believe, in Cardiff. And he did work at Cardiff University and also with the Natural History Museum and National Museum of Wales in Cardiff. He's also worked at in Scotland for the museum in Edinburgh, and done extensive work in China. I can't remember the name of the institution.
Chinese Academy of Sciences.
The Chinese Academy of Sciences, and in recent years he's been a lecturer at Birmingham University, where his specialism is in fossil plants. Tonight, you'll see the title, The Wuda Forest, an earliest example of Permian tropical forest ecosystem preserved under volcanic ash. So I'll leave you to Jason. Thank you.
Okay, well, thank you very much for the invitation to be here, and thank you for the kind words of welcome there, Alan. And also, it's so great to see so many familiar faces. 2 undergraduates from my time. There's probably lots of other undergraduates, or people who have had time at Birmingham University. Actually, another former undergraduate at the back. And also, one person who can remember me from Cardiff University from my PhD times. So it's really nice to have so many people. What I'm going to be talking about today is some research which I've been involved in for about 10 years now, but is finally getting to critical momentum where it's very exciting. I'm going to be talking about the Wuda fossil forest, which is preserved under volcanic ash, and it's something which is really spectacular, and it's rewriting textbooks on a regular basis. It's been known about for not so many years, and it's pretty much showing us everything we knew about Paleozoic coal swamps was generally right, except for when it was very wrong. So it's one of those examples where it's only when you get something with truly exceptional preservation does it help you to explain what the past was like. So this is a site which we would call a Lagerstadt, which is a site of exceptional preservation, in this case exceptional quality of preservation. And I guess most of what you'll be familiar with will be like this diagram here, this picture. This is the classic view of what we understand our coal swamp communities were like. We used to reconstruct them as dark, damp forests. But it's when we look at the plants, actually the canopy was open, there would have been lots of light coming through. And this reconstruction my friend Richard Bateman did in Edinburgh is perhaps the most realistic. It's also the only one I like because it actually shows us what the sediment was like. Usually they just show the plants, the aerial parts. This one's nice because it's done cross sections to show the roots and to show us the plants interacting and the water columns as well. Now we're very familiar with this style of coal swamp. They occurred throughout the Carboniferous of Europe and North America and gave us our extensive coal deposits. Many of you obviously know them here from the Black Country, our industrial heritage. But our views of these have been largely unchanged for many years, and our understanding of them has really not changed very much. And if we look at the late Carboniferous, early Permian world, we have Europe and North America around the central area, the Paleotethys Ocean, and we have the Angaran, the Russian floras of the continental mass to the north, and Gondwana to the south, which was periodically covered in ice, and that ice periodically waxed and waned that gave us our cyclothems and our changing sea level. But what we don't really know is how the floras relate and how the floras of Euramerica relate to the floras of other parts of the world, because we haven't had any continuity between them. They've always been different, and we don't see links to other floras. So this is a diagram that I published with my good friend Chris Cleal many years ago, and it shows the stratigraphic ranges. You can probably see them out, basically going from the Carboniferous through to the Permian, and it shows us the distribution of our coal-forming wetland ecosystems, our coal swamps, as you'll know them from around here. They started in Europe in the latest part of the Mississippian, so the lower Carboniferous in old numbers. And in Europe, they just made it through in a few little pockets through into the early Permian, where they are lost. North America, well, they joined late. They're quite good at joining late. And they finished early as well, compared to Europe. And then we see a loss of these ecosystems. They went out more or less at the Carboniferous Permian boundary. It used to be thought of as an extinction of coal forest plants. We now don't think of it as an extinction. We think of it as what we call an extirpation. An extirpation is a geographical extinction, so it went extinct in one area, but many of those plants survived. And for many years, the Chinese had been publishing plants with the same names and some pictures that said were the same as our Europe and North American plants, and no one believed them. It's only when we actually got hold of the specimens and went and said, gosh, they are the same, that we realized that there was some continuity and these plants were connected, and they seem to have migrated around this Tethyan Ocean through time. So this is the Carboniferous, what's now called the Carboniferous rainforest collapse in its trendiest, latest fashionable term. Is actually a geographical loss of diversity. And we see these things coming through, and we see the same ecosystems perpetuating through the late Pennsylvanian and earliest Permian of North China. And then there's a change, and in North and South China, they're different continents. They join about middle way through the Permian, and those floras migrate between them. And we see them, and we eventually see the loss of these ecosystems around here. So we've got some general information on our plants. We know they're the same, but we're really lacking absolute detailed evidence. We don't really have the quality of preservation in China. And where we do have it, it's just tantalizing, and it's not really showing us more. So this brings us on to the Wuda tuff flora. This is a really spectacular locality in northeast China. So here's Beijing, if you can see the mouse, the red star. That we're over here basically to the west of Beijing and slightly south. And it's in Inner Mongolia, which is Nay Mongol. And it's by the city of Wuhai, and it's a little mining town called Wuda. But it's a typical Chinese coal mine that they've taken everything. And the entire basin is going to be extracted, removing the overlying material in open cast. And this is the scale of the basin as it was in 2010. 2 kilometers side to side there and 10 kilometers. It's actually a much larger area. And we've been very lucky to go and have access to this, working with the local coal mining company to give us access. Now, this is a very complicated diagram just to show you the stratigraphy. We are and the Wuda locality is in the part of what we call the Taiyuan formation. And it's this black layer here is the coal they're extracting. We've got a radiometric date using really precision isotopes of 298.34 million years, plus or minus 0.9 million years. So this is basically down, the accuracy is within 10 to 20,000 years scale of our precision dating. This isn't like the old-fashioned where we do it. And we've got this date because we can date volcanic ashes. The crystals in them can be pure, and we can get thorium and lead dating, really accurate measurements now. So we have the Taiyuan formation in this Wuda locality, which is just up here in the northwest of the North China plate. And in his case, it's basically the earliest Permian. But as you go through the basin, the Taiyuan Formation is what we call diachronous. It's in different ages in different places. This is something I teach my first years in the first few weeks, and it's a bit of a head spin for them. I don't really want to go into that now, but basically the rivers were migrating over an erosive surface, and in different parts, the same fascies, as climate changed, the environments migrated. And as we go over to, in South China here, the Yuzhou coalfield, all of the Taiyuan Formation is in the Permian, whereas in this area here, most of it is in the Carboniferous. So you can't just say this formation is this age anymore, because each region, each basin has a different age. So this is why it's important. These are my colleagues, Herman Pfefferkorn and his wife, Heidi. And this is Wang Jun, who's the big project boss in China. He's now director of Nanjing Institute of Geology and Paleontology. And he's been working on this bed since 2007 paper and a few years before it. And what we have is throughout the basin, we have an ash bed. It's a white, clean volcanic ash. It's called a tuff bed. And that is basically uniform as a marker throughout the coal seam, and it goes everywhere. And this has been the bed that's been dated 298.34 million years. And it's great because when the ash came down, it fell down onto a fossil peat. It was an ecosystem. That peat would have gone through diagenesis to form coal. Plants make peat, diagenesis makes coal. Have to remember that. And in this particular case, everything that was underneath the ash is preserved in situ, in its correct place. You can often get entirely complete plants. We can see their spatial arrangement, we can see their morphology. It hasn't been through the usual food blender of kind of geological processes that I'll come to in a moment. And this is really fantastic. And since it's been first studied, we've now excavated 12,000 1 meter squared quadrats to show where the plants are, and they've all been identified. Something like about 50 or 60,000 plants have been identified. So this is what happens in our thing. So this is a photograph going through it. This is our seat earth on a sandstone. Here's our seat earth, the fossil roots. These are so the seat earth preserves the early pioneering plants, the first ones to grow after the river migrated. This is called Flora 1. Flora 1 that grew and became established. It built up into the peat over tens of thousands of years, and that is our Flora 2. Then we get the volcanic ash, which basically smothers Flora 2. And then once the volcanic ash had actually, after it had settled, the plants started to grow on it again. It was colonized, and that's our Flora 3, an early pioneering flora. And the final stage is Flora 4, where we're getting the whole system is being flooded by a lake, a massive inland lake the size of North China. And that had basically then plants were washed into it, which gives us our fourth floras. So you've got really great succession. So this is a diagram that my former PhD student Layla made for me many years ago. And this is to emphasize that fossil plants are rarely found complete in the fossil record. Think of it as your food processor processing them. Or it's more like a cement mixer. Throw in some sand and some gravel and some bricks, bits of plants, spin it round like a river system, and you don't get complete plants. We have our plants, they're shedding leaves. Here's a food processor. We get bits of ashes, we get log jams, pollen and seeds are lost, fruits are lost, leaves are predated. So this is a real problem for fossil plants. We very rarely get them complete. We only get bits of evidence, so my job is usually to stick them back together, which takes years. But in Wuda, we don't have to do that. The volcanic ash has preserved them in situ. So normally, if this is our fossil plant here, we would normally a paleobotanist would give each different part a different name. So stigmaria is the root, lepidodendron might be the branch, the trunk, The branches might be lepidoploios, the leaves are cyparites, and so on and so on and so on. This is an artificial system. There was only, and we have our taxonomy, the number of species and genera is massive, but the number of plants is probably a lot less. So we're in an amazing situation from Wuda to understand much more of it. And I like to think of it as an analogue for Pompeii. My Chinese colleague called it a Permian Pompeii, or Pompeii. I like to think of it as Pompeii without the Romans, because let's be honest, what have they ever done for us? I think that's please, you are welcome to shout out if you've got that. There is the sanitation and the health and public order. Yeah, okay, so it's like the paleobotany equivalent of something which is truly exceptional. And we don't really have to go and, look at it. And it's on the scale, it's not just one little pocket, a little site with very little exceptional fossils. These fossils are everywhere throughout the basin under this ash. And this ash is covering an area basically the size of most of Birmingham. Probably a good analogue there if anyone wants to think about that one as well. I'll leave it with you. So this is how we do our field work. This isn't just a little tippy-tap geological hammer. We get the excavators in. The coal company want to take the coal. They have a license and a right to extract the coal for combustion. It's giving us all of the sources for the local electricity station. They are very good to work with us and allow us access, and they provide the equipment and the workers and the storage, and they help us move the things around, and it's fantastic. So this is what we do. We go down and clear the surfaces, the bedding planes, using these excavators. We then clear off areas where we're going to samples. We go down to the top of the tuff. We then go and sweep it clean. We then go and put quadrats, 1 meter by 1 meter orientated grid squares, where each one has a GPS location so we can work out exactly where our plants come from. This is just sprinkled on with limestone powder, so it's basically just worked on. And then we go and take out the volcanic tuff, and this is nice because you can see the coal seam above it. The roof shales and the shales above this. And when we keep on working, then we go and basically get on the small tools and take out the blocks. And then my colleagues say, I want that one. If you imagine someone in a wheelchair, I'll have that one. You probably know that joke as well. Quite a good one. We have that entire block, put it in a dumper truck and take it off to our lab to work on. So this is the scale of this operation. And bear in mind, we've got 12,000 of those 1 meter grid squares so far, and it's working. And this is what it's all about. This is a specimen that's about 1.5 meters in length. It's a complete bit of plant. It's only been broken because we couldn't get a bigger piece of rock out. When we collected it, the rest of the trunk was there to go and get the information. So this is the top of this particular one. And on the left is the unprepared fossil, where we just basically cleaned it off and brushed the ash. And on the right is where the really technically talented preparators have dug out and exposed our plants. And so we get really spectacular morphology, the shape of the plants, but we can also go and cut them, and they've often got anatomy. So we've got cellular level preservation. The cellular preservation isn't everywhere, it's just in pockets, but when we get it, it's wonderful, and it allows us to really understand what our plants are like. So this is a Maratialian tree fern. You probably go and think about it as Pecopteris is the usual kind of leaf for this, and you've got lots of these in your museum, Dudley Graham, I would have thought, locked away. So we've got this, and this is one which we've just published. It was in the January issue of International Journal of Plant Sciences, got the front cover of the journal. So this is something that was recently been published, and to go and have a look at the different structures. And here are the different ideas. So here are the individual leaves at the top. Here's another leaf, and you can see the young and the old leaves. We have all the different structures of the leaf. This is the stem, and it shows us these round straws where its branches, actually where its fronds have come out. And previously, we'd seen different kinds of these round structures of the shape of these leaf scars or these frond scars. And previously, we had two really distinctive types, and we've always put them into two different genera. They're not even different species because they're so different. But this one specimen has got both types and shows us that our old idea of these things being two different plants is nonsense, and actually it varies on one single plant. And it's really exciting. So it's allowing us to do it. They also have these really amazing, intricate little short leaves around the bases of the fronds, like some modern-day Maratialian ferns that seem to be stopping insects. These are the reproductive organs. We've got the leaves. They have their spore-bearing sacs underneath them. And we have these different structures. You can see them all in attachment to the same structure. So everything is connected to everything else. And many of the things have our anatomy. And even these ones, we could then actually go and get the the spores out of them. And that allows us to reconstruct a single specimen like this with many hours of painstaking reconstruction. And it turns out this is the first actual species of this ever to be reconstructed. Before, we've done a general composite. Oh, they have this and this and this. And that's really hiding diversity. So this is much better. And in this particular case, it's showing us really what's happening and these various different structures. And as you can see in here, all of the different parts of it. So here's the crown, here's our little vegetative leaves at the base here's our fronds. And these ones at the top of the crown you saw with the frond scars is here. And these are the roots that are coming down from it. But we don't just get one of those, we've got lots of species. Here's another one. We don't have the whole plant, we've just got the leaves, so we don't always get everything. Walk into a forest, you don't always get the plant. Sometimes you get branches and things that have come off. And this is one where we've had a look at it, and this is another one where we've reconstructed it. And I've shown this one again. It's published January or February. This has been published in the last few months. And this is why we've shown this one. These are its spore-bearing units. This is looking down. This is them looking across. And this is the sporangia. The spore body sacs are in here. And they have the leaf coming down to envelop and hide it. And inside that, we have all of these individual spores. And here's the sacs full of spores. And the level of detail is, you get as good as the garden centre, basically. So it's really, really spectacular. We get some really weird, some unusual plants. This is one called Wuda phyton. It's a climbing plant. Climbing plants in the fossil record are really incredibly rare, and we're lucky to have them. And this one is really small, so it doesn't photograph well, but basically it's lots of very thin, slender shoots and lots of hooks and leaves with little sticky buds on them. And when we looked at this one plant, it was really confusing. It's got 4 different kinds of leaf in different parts of the same plant. And if we'd have found those just loose, those individual leaves, we'd have put them into at least 4 species, probably 2 different genera. Actually, it's all belonging to one species. So it's showing us what biological variation is like and making us think more like biologists. And even better than that, this is a reconstruction of Wuda phyton, our climbing plant. We can look at where it occurs in the volcanic ash. Here's our 66 centimetre, I just put it at 600 millimetres here. And here's the profile and the sections of the sections. And we can work out how much of it occurs at each different level. And then we can also predict onto which plants it's growing on. We do have it growing on trunks. We can't tell which ones they are. But because we know how big all the other trees are, if it's occurring in the top part of the ash, because when the big trees fell, they fell last, and they occur only in the top part of the ash, or their crowns do. Their roots are usually in the base. We can tell that these things are growing on trees up to 10 metres tall. So we know the short ones are growing on the tree ferns, like the ones you've just looked at, but these ones here at the top are growing on Cordytes plants. And now you may be familiar, Cordytes is a group of plants probably believed to be closely related to conifers in the broadest sense, a sister group or an ancestral lineage to conifers. Now, this is something where we've had really exciting work, and this is something which has been preoccupying me for the last few months, trying to get this finished. And this is how a group of plants called cycads, the photograph in the top is from a botanical garden, where this is a modern-day cycad, and these are quintessential living fossils. They're plants which, you know, we have a massive diversity of them in the Jurassic. We have a few of them in the Permian, but we didn't really know where they originated. A traditional understanding is that they're meant to be closely related to medullosum seed ferns, so things like Allothopteris type and Neuropteris type leaf foliage. And that's basically, that idea has been around for about 125 years or so, and no real solid data. Modern day biologists conceptualize it like this, but they have no understanding of fossils or evolution, and I could spend hours taking that to pieces, but basically modern day biologists don't have an answer to this, where cycads evolved from. So we know that we have some fossils previously that we think we've got bits of cycads. We've got some of their fertile leaves in the Permian. But we haven't got their plant, so we don't know what they look like. So it's been tantalizing. Everything we've known about them is fragmentary bits and pieces until you go to Wuda, where we've got them. Not just one or two, but we're so far about between 12 and 17, depending on which versions of the numbers we look at. And here's an example on the left. This one here is a large trunk. There's a piece missing, but when you break the rock, it's on the other side. This is just a field photograph, so we know it's connected. And this is the crown up here, and these are the big leaves of the cycad plant. Here's another one, here's where the stem is, and here's showing the crown of leaves. And here's another one, which is the trunk of this and the leaves. So we've been very lucky to get complete fossil cycads, and this is really spectacular. But unlike modern-day cycads, their leaves are divided, they're like a fern pinnule, they're very, very segmented. But our one we have, that we're calling Eocycas, at the moment, this is our reconstruction, the best piece. The leaves are entire and long strap-shaped leaves, and modern-day cycads don't do this. But everything else about this plant is the same as cycads. In fact, if you were to go to a botanical garden and see this thing growing, other than the leaves, you would think it was a modern-day cycad. And this is like the Rosetta Stone. This is really complicated, and this is one of the crowns coming through here. Crown coming through here. So this is the base of the crown, and these are the leaves coming down. And this is actually another one here on another layer of the sediment. So it's quite confusing because it's in the ash, one has fallen this way and one has fallen the other way on top of it. So they were, living close by each other. And this one has all its fertile units. In this case, it has its ovules or its seeds, and the seeds are attached to a fertile leaf. And this is what they look like. So here's the stem. Here's an artist's drawing of the actual specimen of the leaves. And these bits here are where the bits have been eaten, so the leaves. And this yellow structure here is a fertile leaf. So basically, the leaves, they have vegetative leaves for photosynthesis. Then they have a fertile leaf, which has been evolutionarily adapted, and it basically is to bear a structure for bearing the seeds. And when we have a look at this, how this bears the seeds are very, very similar to modern cycads. Again, if you were to see them in the fossil record, there's slight differences, but the same. So here are some photographs. Here are some of these beautiful round seeds, and here's the seeds and the parts of the cone. Here's some of basically these lovely little leaves, the sporophylls they occur on. This is the fertile leaf with the seeds attached. They basically had to dig away to go and get their morphology and structures. The downside is we haven't got anatomy in the seeds of these ones. That's really something. I've been working on anatomically preserved seeds for 30 years. That's really what I wanted. But we've got everything except for the bit I really wanted. There's no justice. We just need to keep looking harder. And this is, so this is another specimen. So this is one specimen, one of our trees. Basically, this specimen, it doesn't form a cone. But in the same assemblage, we have one cycad which does form a cone. And modern day cycads, some of them form cones and some of them don't form cones. And we've got both of those morphologies from the onset. And here's another one. And this one has two beautiful seeds at the base of this highly intricate fertile leaf, which looks very similar to modern day cycads and cycadaceae. And this is our basically the structure of our fossil. Now, we're lucky with these. We know absolutely they're cycads because the seeds, the reproductive organs are cycads, but they're a little bit different. But their leaves are very uncycad-like, but their stem anatomy is definitive cycads. And this is an example. This is what a single specimen as we found it in the ash. And these structures here are where the individual leaf or traces come out of it. And it's basically a cast of the part of the cone. And this is basically then you go and cut it through in a section, so it's cross-section made through here. And these black things here are the xylem, the wood. And in between it, these are the leaf traces where individual leaves come out. And we've got some really spectacular tissues. And they are, this axial parenchyma is definitive and distinctive of cycads. We have these sclerotic tissues that, again, characteristic of cycads. But we get this one really bizarre thing in cycads. They just do it, they're just weird. There's no other way to describe it. When normally plants have a branch coming out, the branch, it comes out from the center of the plant. This is where the vascular tissues come in, the water and food come out. And they usually come out in a straight line from the nearest side. Except for cycads that do it that way, but they also have plan B, where the leaf traces on the other side go all the way around the stem and come out. So it's actually got, the leaf traces come out on opposite sides. One goes out directly, and the other one goes all the way around the houses and comes out the opposite side. We have no idea why they do this, but they all do. And I really struggling to work out the function or the adaptive significance of this. But it's just remarkable. And we have exactly this in our Eocycas, and we have some of them that go out directly, and other ones that go all the way around the houses. So again, this is a cycad. So this is what our reconstruction is like, so it's slightly yellow here. So this is our reconstruction of our cycad of our plant with our crown and our cones in here. Sorry, our non-cone are basically our fertile leaves. And this is what the fertile leaves look like, usually with 2 to 4. And this is our reconstruction of it in the forest, because obviously we know all the other plants that live around it. So this is the next thing we did. We looked at the paleoecology, because fossil cycads, there must be something unusual about them, why we don't have more of them. So out of about 50,000 specimens identified, from 12,000 1 meter quadrats, only 16 trees occur of cycads in 3 little clusters. And those have been plotted here. They're all quite close to each other. We've also got the leaves were attached to our cycad trees. We've only got them in the trees and about 100 loose ones out of about 50,000 specimens. These are really not common, and they were living in clusters. So in other words, you're going to have to do a really big sample to go and get one. And our sampling has got that level. And have we got sampled, 50,000 leaf specimens from the South Stafford's coalfield to see if we've got any cycads? I don't think we've got to that level of and every one of them being identified. So we just haven't done enough of it to go and get it. So my Chinese colleagues have been very studious. And their distribution, they're not everywhere through our forest, they're just in these little pockets, and they're growing in groves. Modern day cycads are exactly the same. They grow in groves with a few little trees here and there. So that's good. Now, the final one I want to talk about is the unpronounceable plant group called the Noegarathiales. We'll just call them the Noegs. It's a ridiculous name, but it has taxonomic priority, so we're not allowed to change it just because it's ridiculous. We're stuck with it. Okay? These have been known since the early 1900s. We didn't know what they were. They were a taxonomic football. Different researchers have kicked them and said they're this group of plants, oh no, they're this group of plants, and no one scored a goal in all that time to work out what they are. It's only with this specimen called Paratingia in Wuda. We've now got another one called Tingia, and this is the specimen. It's about 2 meters long. It's the key specimen. It's an entire Noegarathialian plant, and as well as having the morphology, we also have the anatomy of the different parts. We can put all the pieces together, and this is our Rosetta Stone. Trees are typically 4 to 6 meters tall, naked trunks. We've had lots of different names for them, but we've just not known what they were or what they looked like. We've only ever found pieces. This is what the leaves look like. These are basically the leaves. They're flattened in 2 dimensions, except for they have another set of leaves that are at 90 degrees to these standing upright. So they're basically in a cross shape, but coming out like a frond. They have the 2 big leaves flattened and the little leaves. They have really, here's some of the anatomy showing our vascular structures, and again our rays and our xylems and our different tissues. We also have the anatomy through the rachis, which is this part through here, to go and show us what shape the vascular system is. That shape is systematically distinctive. It basically, each group of plants has its own unique features or things we can recognize. But we also have their cones that are complete and preserved in the ash. Here's various different sections of them. And the previous ferns I talked about had their spore sacs. The cycads have their seeds. These plants do it 2 ways, pretty much. They have their males are called microspores and the microsporangia. And these are the little structures in here, millions and millions of microspores. But as well as that, they have what are megaspores, and they're megaspores, these things here called the ME, and they're a seed-like structure, but in a fern. They're not actually a seed, they're just the size and shape of a seed, and they're just born in a way. And this is something that's really, really unusual. And each of these, instead of having lots of megaspores, has a single functional megaspore in it. So we call these monomegasporic. And strangely, these are rather than organized randomly in a cone, they're in vertical rows, which doesn't really make much sense. Plants usually do things in helixes. This group of plants, everything else is in helixes, except for these female structures, which are in vertical rows, which again, rather confusing. This is going a section through the actual stem. In this particular case, this is going through the rachis of the leaf, and it has what we call an omega-shaped bundle, like the Greek symbol, an omega. And this also occurs in the trunk of the plant, and this basically is telling us it's a very weird group of ferns. How they've evolved their reproductive cone. It's not a cone, it's a pseudo-cone. Cones do things helically, or except for those that do things decussately. And the cones always have a rounded central vascular tissue. These ones, because they've evolved from a fern, have it as a fern-shaped omega trace, and they've just done it completely different to every other group of plants that's out there. Again, they've evolved a cone, but from a very different starting point. And you look at them, you think, oh, that's a cone, it's just a cone. Only when you get the anatomy, you realize it's anything but a cone. It's a pseudo-cone. It just looks like a cone. Evolution working at its best. This is our reconstruction of what it looks like. To the right is a diagrammatic reconstruction. To the left is basically us showing what all the different pieces look like. So here's your trunk coming through it. and you have some of these leaves coming off, or basically the frond coming off it. Then you have the cones in this zone. Then at the top of it, the cone doesn't occur at the apex. It has more leaves coming down. And as the plant grows older, the leaves are basically shed and dropped off. And this is what the cones look like. They're really weird organization. And this is the various different structures of the leaf. Okay, so because we have the anatomy and the morphology of these plants for the first time, I did an evolutionary analysis where I put everything together to try and work out, were these ferns, they've been grouped with about 10 different groups of plants have been proposed as being their closest relative. And we did the analysis and we found out that all those plants that were suggested as being their relatives weren't. And they were actually the closest relative of seed plants. So we previously thought they were a group of ferns, but they turned out they were ferns which are closest origin to seed plants, so in the ancestral lineage, which was something really exciting. And it's something which we then went to publish in the Proceedings of the National Academy of the Sciences in America. So after 121 years of being known, we finally worked out what they are. So this is why this assemblage is so important to help us sort out what's coming on. My final point is, what we're doing now is we're actually looking at the quadrat data, our 1 meter by 1 meter squares across the forest. We're plotting what species occur where, and we're looking at community and ecology, and we're looking at which plants co-occur, and we're looking at their spatial distribution, and we're looking for the plants that are growing and occurring together, and doing some really exciting ecology work. And I think this is something which is going to be taking us many years in the future. And this is something which we expect to be working on this for many years going into the future. So that's something that I'm doing. And also, this is also project work which I've got my students working in. Now, the final part is what we're doing from now, our future studies. In conclusion, our Wuda flora is something which is world-class, remarkable, and really exciting. It's giving us an unrivaled view into a Permian terrestrial ecosystem, and it's revolutionizing our understanding of many different things. In particular, it's showing us the links between the Euro-American and the Caucasian flora, and how these are responding to climate change. And in particular, when it got too hot in Europe and North America, these plants died out, and by then they had migrated around to China, where it stayed warm and wet. But once those climates dried out in the middle Permian in North China and the late Permian in South China, these floras went extinct then. It's giving us amazing information about the origin and evolutionary relationships of major groups. It's giving us great information about ecology, paleoecology, and understanding our fossils as once living species in a way we just can't do from other localities. And the final thing out it is something which I think is giving us great pleasure to do and giving us lots of curiosity-driven answers to really understand much better than we ever have done before. The site has in the last few years gained international recognition. The International Union of Geological Sciences has designated it as one of its top global sites. UNESCO plans are in, UNESCO links are being planned at the moment, which is why we've got to speak very nicely to Graham over here to get some advice and recommendations. And before the pandemic, there was going to be a new museum open to showcase these fossils, but then in the pandemic it got mothballed, and we heard about 6 months ago this museum was back on the table, and so we got really excited. And I was used to a British style or American style of we'll build a museum in 5 years, we'll then start to put the exhibitions together. No, it's going to be opened by summer 2025. So not just built, not just the exhibitions and populated and the visitors numbers, but it's going to be delivered. So I think Graham's now looking forward to seeing if we can get him out there. So this is something that's really exciting, and this has led to so far about 15 to 20 PhDs, and in the region of, a dozen or more master's student projects as well. And it's something which I hope will continue into the future. This work has only been possible from the work of Professor Wang Jun at Nanjing Institute of Geology and Paleontology and his team. So Li Dan, Wei Ming Zhou and Sun Wenjun, and my good old colleague Wang Xijun. We have to acknowledge the work of many tens of people and hundreds of people over the years doing this site excavation. It's also the work of Herman Pfefferkorn and a group of the Czech Paleobotany and Palynology Consortium. I tend to think of as the Czech mafia, because when they get their hands on something, you're not getting it back. And also this work has been worked, and particularly for the cycads, with Mario Coiro and Leila Seyfullah, University of Vienna, and now in Senckenberg in Germany, and my colleagues in China. So I will draw a halt there. So thank you.
And that's a very opportune moment for me to say the formal vote of thanks, Jason, and to say how opportune it is that here you are delivering the second presentation to the Black Country Geological Society in our 50th year, 2025. The second presentation that was done in our first year was on the South Staffordshire Thick Coal. How appropriate.
Who was that by?
That was by Basil Poole.
Okay.
He was the senior partner with Johnson Poole, the local mining consultants. Not a scientist, a very different take on that. How you mined it and how you got the best value out of it and how you stop people from killing themselves.
It's still relevant.
It's very relevant. So, with that, could you all join me in thanking Jason with the usual way?